Artificial intelligence is now capable of generating novel, functionally active proteins, thanks to recently published work by researchers from Chalmers University of Technology, Sweden. Proteins are large, complex molecules that play a crucial role in all living cells, building, modifying, and breaking down other molecules naturally inside our cells. They are also widely used in industrial processes and products and in our daily lives. Protein-based drugs are very common. The diabetes drug insulin is one of the most prescribed. Some of the most expensive and effective cancer medicines are also protein-based, as well as the antibody formulas currently being used to treat COVID-19. Current methods used for protein engineering rely on introducing random mutations to protein sequences. However, with each additional random mutation introduced, the protein activity declines. Consequently, one must perform multiple rounds of very expensive and time-consuming experiments, screening millions of variants to engineer proteins and enzymes that end up being significantly different from those found in nature. This engineering process is very slow, but now the researchers have an AI-based method where they can go from computer design to working protein in just a few weeks. The new results from the Chalmers researchers were recently published in the journal Nature Machine Intelligence and represent a breakthrough in the field of synthetic proteins. The researchers have developed an AI-based approach called Protein GAN, which uses a generative deep learning approach. In essence, the AI is provided with a large amount of data from well-studied proteins. It studies this data and attempts to create new proteins based on it. At the same time, another part of the AI tries to figure out if the synthetic proteins are fake or not. The proteins are sent back and forth in the system until the AI cannot tell apart natural and synthetic proteins anymore. This method is well known for creating photos and videos of people who do not exist. But in this study, it was used for producing highly diverse protein variants with naturalistic-like physical properties that could be tested for their functions. The proteins, while used in everyday products, are not always entirely natural, but are made through synthetic biology and protein engineering techniques. Using these techniques, the original protein sequences are modified in the hope of creating synthetic novel protein variants that are more efficient, stable, and tailored towards particular applications. The new AI-based approach is of importance for developing efficient industrial enzymes as well as new protein-based therapies such as antibodies and vaccines. The researchers say that accelerating the rate at which they engineer proteins is very important for driving down development costs for enzyme catalysts. This is the key for realizing enormously sustainable industrial processes and consumer products, and their AI model as well as future models will enable that. The next step for the researchers is to explore how the technology could be used for specific improvements to protein properties, such as increased stability, something which could have great benefit for proteins used in industrial technology.